The digestive system includes the gastrointestinal tract, mouth to anus, the glandular organs. This system serves to transfer organic molecules, salts, and water from the external environment to the body's internal environment. Most of the food taken into the mouth are large particles containing macromolecules such as polysaccharides and proteins. As such, they cannot be absorbed by the intestinal wall. They must be dissolved and broken down into much smaller molecules. This process is named as digestion. Digestion is accomplished by substances called enzymes produced from the digestive glands. The enzymes are biocatalysts in the food breakdown process, the process of digestion. Mouth. In the mouth, digestion starts with chewing. It breaks up large pieces of food into smaller particles. They can be swallowed without choking. It is accomplished by teeth, tongue, jaws, and saliva. Chewing is controlled by the stomach nerves to the skeletal muscles of the mouth and jaw. Rhythmic chewing motions are reflexly activated by the pressure of food against the gums, hard palate at the roof of the mouth and tongue. Saliva. Saliva is secreted by three pairs of exocrine glands, namely as parotid, submandibular, and sublingual. The daily secretion of saliva ranges from 1000 to 1500 milliliters. It contains the organic substances amylase and mucin. Salivary amylase or phthalate can act on starch. It converts cooked starch into the disaccharide maltose. Mucin is a glycoprotein. It helps in the lubrication of food. The lubricated swallowable form of food is called a bolus. The salivary secretion is controlled by reflex activities. Swallowing. It is a complex reflex activity. It is controlled by the swallowing center in the medulla oblongata. During swallowing, the soft palate is elevated, the larynx gets raised, the tongue forces the food back into the pharynx, the epiglottis closes the glottis, and the food slowly passes into the esophagus. The esophageal phase begins with relaxation of the upper esophageal sphincter in the esophagus. The food is moved toward the stomach by a progressive wave of muscle contractions. Process downward to the stomach. Such waves of contraction in the muscle layer surrounding a tube is known as peristaltic waves. In the esophagus, one peristaltic wave takes about 9 seconds to reach the stomach. Due to peristaltic waves, swallowing can occur even while a person is upside down. The stomach. It is a white chamber located below the diaphragm. The size and shape of the stomach depends on the blood inside it. The stomach volume during feeding may increase up to 1.5 liters. Stomach primary contractile action will produce peristaltic waves. Each wave begins in the body of the stomach and proceeds towards the pyloric region. The initial wave influences the muscles to close the pyloric sphincter, a ring of smooth muscles between the stomach and the duodenum. The inner wall of the stomach is lined with gastric glands. There are nearly 40 million glands engaged in producing gastric juice. The chief cells of the lining of stomach secrete enzymes, and the parietal cells, oxantic cells, produce hydrochloric acids, strong acid, to create acidic medium for enzymes. The enzymes of the stomach are the pepsin and renin. Pepsin is secreted in an inactive precursor form known as pepsinogen. The activity due to hydrochloric acid converts pepsinogen into pepsin. Pepsin hydrolyzes the protein into short polypeptide chains and peptones. It is most effective in an acidic environment. Protein, pepsin, polypeptides plus peptones. Acts on soluble milk proteins, casinogen, and converts it into insoluble casein. In the presence of calcium ions, casein is precipitated as insoluble calcium casein compound curves. Repeated peristaltic valves in the stomach helps to soften the food. The frequency of contraction is determined by the basic electrical rhythm and remains essentially constant. It is also aided by neural and hormonal influences. The food leaves the stomach in the form of chyme and enters the upper small intestine in periodic intervals. The small intestine. It is about 5 to 7 meter long. It is divided into three segments, namely as the initial short segment, duodenum, the jejunum, and the longest segment, the ileum. The food is propelled down into duodenum due to peristaltic action of stomach wall. The pyloric sphincter located at the junction of stomach and duodenum regulates movement of chyme. The food in small intestine is mixed with three juices, namely bile juice, pancreatic juice, and intestinal juice. Bile juice. It is a brownish green alkaline secretion of the liver. It is stored in the gallbladder and poured into the duodenum via the bile duct. The bile contains water, mucus, inorganic salts, cholesterol, and bile salts. The bile salts emulsify fats and help enzymes like lipase to act upon fats during emulsification. The bile salts converts bigger fat particles into smaller fat globules. Pancreatic juice. It is an alkaline fluid, pH 7 to 8. It is transported through the duodenum to the pancreatic duct. It contains water, mineral salts, and a variety of enzymes like trypsin, chymotrypsin, which are secreted in the form of inactive precursors. Trypsinogen and chymotrypsinogen, the precursors are activated by enterokinase of the intestinal juice. The amylectic enzymes amylase, the pancreatic lipase, trypsin, carboxypeptidase, and nuclease. The enzyme trypsin hydrolyzes protein into polypeptides and peptones. Proteins, trypsin, polypeptide plus peptones. Chymotrypsin hydrolyzes peptide bonds associated with specific amino acids like phenylalanine, tyrosine, and tryptophan. It results in large peptides, protein, chymotrypsin, large peptides. Carbo Carboxypeptidase is an exopeptidase. It attacks the peptide bonds at the carboxyl end and the polypeptide chain, resulting in tripeptides, and amino acids. The pancreatic amylase converts starch into maltose. The lipase acts on emulsified fat, triglycerides, and hydrolyzes them into free fatty acids and monoglycerides. Monoglycerides may be further hydrolyzed to fatty acids and glycerol. Intestinal juice, maltose, maltase, glucose plus glucose. sucrose, sucrase, glucose plus fructose, lactose, lactase, glucose plus galactose. Ditripeptides, peptidase, amino acids, nucleotides, nucleotidase, nucleoside, phosphoric acid, nucleoside, nucleosidase, nitrogenous base plus sugar. Absorption and assimilation. As a result of digestion, all macromolecules of food are converted into their corresponding monomeric units. Carbohydrates are broken into monosaccharides such as glucose and fructose. Proteins are hydrolyzed into amino acids. Lipids get broken into glycerol and fatty acids. The simpler organic molecules along with minerals, vitamins and water enter into body fluid through the villi. Villi are found in small intestine. The villi are small microscopic finger-like projections. Each phallus is an absorbing unit consisting of a lacteal duct in the middle surrounded by a fine network of blood capillaries. While the fatty acids are absorbed by lymph ducts, other materials are absorbed either actively or passively by the capillaries of the villi. From the lumen of the alimentary canal, absorbed food material are carried to the liver through hepatic portal vein. From the liver, materials are transported to all other regions of the body for utilization. This conversion of food into energy and cellular organization is called assimilation.